Good morning, and welcome to this Baltic Breakfast. It's about polluted marine sediments. My name is Ellen Bruno. I work at the Stockholm University Baltic Sea Center. We arranged this breakfast to highlight important areas or important research in areas that may be important for policymakers, and we hope that it can be useful for them. And I would just have to say, sadly, that normally we would have two researchers here today, but Divya Pal has had problems getting back from India, so she won't join us, unfortunately. But Jonas Gunnarsson, you'll hear, and you'll cover her part as well. And as always, we do one half-an-hour presentation, and then you have your question for 15 minutes. So keep them and send them for you online through email or Slido. You can find on the web. Okay, so why do you want to talk about polluted marine sediments? Well, for once, they are very common. We have been polluting our waters for many, many years through paper industries. We've done steelworks and other industries. And of course, many of these pollutants end up in the marine environment. Here they can stay for a very long time. They can also keep polluting surrounding environment through different ways of up in the system again. And on land, normally we would dredge, or we will remove those sediments, we put them in the landfills, more or less safe, and keep them there. But in the marine environment, it's more difficult. We have a lot of water in the sediments, and we have to take care of them somehow. So normally we have been dumping them in deeper waters. We done a breakfast and a policy brief about that a couple of years ago, if you want to read about the hazards of that. There's been some critique against it. I think those of you who live in Stockholm follow the discussion on dumping sediment from Ropsten in Kanholmsfjärden. So we want to investigate what other methods are there. Can you just keep them, maybe treat them somewhere else? And for that, I have the perfect professor, I think, to help us with that. Jonas Gunnarsson, you are a professor at the Department of Ecology, Environment and Plant Sciences, exactly, at Stockholm University. And you've been doing this for a long time, and I think you have a lot of knowledge to share. So please, take it away.
Okay. All right. So good morning, everybody. And first, a big thank you to Ellen and to the organizers. And then, ladies and gentlemen, colleagues here and online, and all our students also. It's a pleasure to be here at this now prestigious Baltic Breakfast seminars. So it's an honor to be here. And let me take some 30 minutes of your time to share some key insights that we have gotten on these polluted sediments, and over more than 20 years. So I work as a professor at the DEEP, is easier to say than the long name, and I work as a teacher, and I work also as a researcher. And before I start the presentation today, I just want to pay a short tribute to three key persons who have been important through my career. And first we have Jacques Cousteau, who inspired me to become a marine biologist. And then we have Rutger Rosenberg, who was my coach during my PhD time and introduced me to benthic ecology and sediments. And then I have my father, Staffan Gunnarsson, who is 92 years old and still going strong, and who is here in the public and listens to us and to me for the first time. So happy you are here. And what I will talk about today is mostly the results from a recent research project we've been working in this harbor in Oskarshamn for about 10 years, and with funding from the Swedish Geotechnical Institute in the TUFO program, and also with funding from the Swedish EPA. And it's not only me, we are a big team, and I would like to acknowledge, and many of them are here today also, and also online, and unfortunately Divya, our wonderful colleague from India, she cannot be here with us today, but she's with us online. And we've had many students, mostly, those are the guys who've been doing most of the work. And then we've had postdocs, PhD students, 12 researchers, help from the Marine Research Laboratory at our department and from the ASCO staff. And it all comes to the sediment. And like you were saying, Ellen, the common remediation that we do with our sediments is that usually that we try to dredge the sediments and take them away from the environment and place them somewhere else. And this has some benefits. It will help to remove the contaminants from the polluted site, but it also often has a lot of problems. It can cause resuspension, and it can cause the contaminants that were safely buried in the sediment that come up instead and expose fish and other animals. So the key question is it really always better to remove the sediments, or maybe sometimes it's better to leave them there in place? And this case study we've been working on, the Oskarshamn Harbor, is a classic type of heavy industrialized and polluted harbor. And we used to have the Kopparverket copper factory here, who has released tons and tons of copper and other chemicals and contaminants. So the sediment is heavily contaminated with metals. And also we found out that it had a lot of dioxins. And because of the dioxin levels that were so high, it was one of the highest hotspots in the whole Baltic Sea. The Swedish EPA and the municipality decided that we should try to dredge it. And the whole inner harbor here has been actually dredged, and the sediment has been taken up. I realize here the light is not so easy to see. It's a little bit too light. I don't know if you can, Jens. Anyway, I hope you see. So we have, they've used different dredgers, suction dredgers. I think this is better. And then all these land masses, like 700,000 cubic meters, like the whole Globen, if you in Stockholm, if you can imagine that amount, with has been transported to a huge landfill that was constructed nearby. And so when we do sediment remediation, we can do different ways. One way, the most easy way, is to do nothing. Basically, we do natural monitored recovery. So what we do there is that we leave the things there, but then we have a monitoring program, and we try to follow over time what happens. The other way is to dredge. And another way is what we do also is what we call capping. So basically you put isolation sheets with different materials like sand, and you put maybe 1 meter of isolation materials to try to keep all the contaminants on the bottom. And another method is this, what we call thin layer capping with sorbent. So this sorbent, often it's activated carbon, a little bit hard to see on the slide here, but there is a top of activated carbon placed here in just a beaker to show you, and then the animals would biotubate this material and mix it into the sediment, and the material would then, the carbon will then sorb the contaminants. And why activated carbon is because we also call it activated biochar, and that's, when we get poisoned, you go to the hospital, you will drink a glass of water and take some biochar pills, and we do the same, but try it in the sediment. So we pulverize activated carbon, and we try to put it on top of the sediment. And why is this just activated material so good? It's because it contains a lot of pores, micropores. Here you would see some small pore. So it's a pore structure, and these pores give it a huge surface. So hard to understand this, even for me. 1 gram of activated carbon is about 1,200 square meter of a surface. So 30 times 30 meter of a surface inside 1 gram. So that makes us think a little bit. And of course, this activated carbon is used a lot for water treatment. So this is an extension of the method to try to use it for sediment treatment. And we started it in Norway for a few years ago with my colleagues from NIVA and the Norwegian Geotechnical Institute. And what we did was that we took tons of activated carbon and put them inside of a big boat, a suction dredger barge, and we mixed activated carbon with clay that came from a clean site and added salt. And then we pumped this at a 30-meter depth and at 90-meter depth. So it was a way to test if the method works, if it's possible to do it. And you can see here the materials on the bottom taken with a sediment profile camera. This is the activated carbon. This is a reference that has nothing. So the material gets mixed into the sediment. And the idea with this activated carbon then, that is all these white spots here are pollutants, and they don't like water, so they will try to sorb to sediment particles, but they will sorb preferentially to this activated carbon, go into the micropores. And we've seen in many studies that we can decrease up between like 60 and 90% of the uptake of these contaminants in fish and in sediment biota. And the question is that when we did it in the beginning, what effects do we have on the benthic community? So we measured in these fjords in Norway, even if you can read the details, just to get an idea on the y-axis here, what you see is the fluxes, the release of dioxin from the bottom to the water. And then we have time, and we have different treatments. So we compared, we have reference, we have limestone only, clay, and clay plus activated carbon. And you see the one with the red arrows, that's really where we have the lowest release of this dioxin. So it worked very well in terms of retaining the dioxin. So it worked well for contaminant sequestration, but we got a lot of problems on the benthic community. So the animals were not happy at all with this treatment, so giving us the orange light. And we could stop soon the presentation if I just make it in short here, meaning that in the Oskarshamn harbor, what we did was that we found a new material that doesn't cause these problems with the negative effects on benthic fauna anymore, and that is still effective. So we've gone from the orange light to the go light, basically now to we are ready to implement this technique in the field. And when we started this captive project, there's a lot of questions for entrepreneurs who want to work with this method. You want to know, okay, can we remediate the outer harbor of Oskarshamn that has not been remediated yet instead of dredging? And so we studied the outer harbor. We studied in the lab. We looked at what type and size of activated carbon sorbent we should use, not to cause any negative effects. We also studied how to apply this material. Should we do it with the barge and the ships like we had in Norway, or other ways? What dose do you need to, how much do you need to add? And what happens when the sediment gets resuspended? Is it still effective? How long time is it lasting? Do we need to repeat it every year or never? And what are the costs compared to dredging? So all these questions we addressed in the captive report. And there is a report coming out now in spring in Swedish, unfortunately, but many of you can find it soon online. And so in the captive project, we tried then different particle size, different types. We also tried fossil activated carbon. We tried biochar, like biomass activated carbon. We found that the biochar is much better. And we tested 10 activated carbon, 8 biochars, and we can basically divide them into powdered, or that we call powdered activated carbon, or PAC, or granular activated carbon, or GAC. And the powdered one is under 150 micrometer, very small particles. The granular one are more up to 5 millimeter. And the powdered one works best in terms of sorption efficiency. but it tends to resuspend in the field. It also causes these negative effects on the animals, whereas the granular activated material has no negative effects on the animals, but it's not so effective. So we were like between these two. So we were thinking, can we find something in between? Can a particle size between the PAH and the GAC work? And in the literature, there's been a lot of studies in the field with this method. And you could see two ones done in the US there. They had 75 to 300 micrometer, and they had no effects of, no negative effects of the material. Whereas all the one here in red, or the one we did in Norway, and they've done in the Netherlands also, we had finer materials, and it caused negative effects. So we don't want to use this powder activated carbon. In the captive project, we continued this, and you cannot read this, but you can see the two colors, the red and the green. And basically all the red studies there are studies that have shown negative effects when you use the PAH. And whereas when you've used the GAC, the granular activated carbon, you see we don't have any effects. So what we found in our studies, we confirmed what other had suggested, is that this powder activated carbon, when it's taken up in the animals, it does not only take away the contaminants, but it also binds all the essential nutrients, the food. So this becomes like a leaning, like bantningskur. How do you say that? A diet, yes, for basically a slim regimen for the benthic fauna. So that's not what you want to get. And what we thought about with Robert, my PhD student, former PhD students who is here with us, we thought that, okay, most of the benthic fauna, they actually feed on particles that are below 100 micrometer. So if we can go just above 100 micrometer, but below 300 micrometer, and test in this range, because above 300 micrometer is not so efficient anymore, and below 100 micrometer, that's where you would get the negative effects. And indeed, you can see from this happy worm here, so this actually looks like it's a transition of 1 second now on a slide, but this took like 3 years to find in the lab. We found a material between 100 and 250 micrometer that is quite effective, and that does not give any negative effects. And then the activated carbon, it's hard to dose it as it is. You want to have a carrier material. So we also tried different types of materials, and one of them that we've been working with is material called polonite. So it's a mineral material, so it's a calcium silicate-rich calcium carbonate, or we can call it calcium silicate, that is mined in Poland. That's why it's called polonite. And it's often used in wastewater treatment to trap phosphate and also to trap some metals. So what we did was to mix polonite and activated carbon to see if that could be make the treatment even better, because the polonite could also be helping to sorb many of the metals. And this is how we usually been working. So you see two maps up there with Oskarshamn Harbor. And we've been taking samples, especially in the outer harbor at different places, using different type of equipment. We have like tube cores, and we have a large sampler that was specially built for this project with contributing funding from the Baltic Sea Center. You see one of the Gotland ferry that comes into the harbor, and that has been one of the problems because they resuspend a lot of materials when they come in. So with this big sampler, we can take like a piece of cake, if you want, from the bottom and bring it into the lab, and we bring it by boat to Askö laboratory. And in the Askö laboratory here we have Johan Wikström, also one of my former PhD students. I think he's here today also. And we did an experiment where we added either just activated carbon or just polonite, or activated carbon and polonite, or had controls. And not having time to give you all the detail, I can just tell you it works. You can read this paper. All the green arrow here showing that the method was efficient, meaning that we could decrease the dioxins, we could decrease the metal uptake in worms, we could decrease the PCB, we could decrease also the metal bioavailability in passive samplers. And we had some red arrows, which is that the pollenite in these first experiments was not working very well. Then we had to reduce the dose of the pollenite, and then we came up to a better, now ideal system. So the conclusions of this is that then the granular activated carbon, 100 to 250 micrometer, it works. We can decrease the bioavailability up to 95% of many of the pollutants. We also calculating the dose. In the US, they often use too much of it, that causing negative effects. We found that you just need to increase about 1.5% the own carbon content of the sediment, and then it is efficient enough. And that corresponds to 600 gram granular activated carbon per square meter. But you have to remember that this has to be also carefully evaluated from case to case. We have to know how much carbon there is in the sediment, what type of contaminants, and then you can adapt the sorbent. For the pollenite, we also found that actually adding a 600 gram pollenite with 600 gram of activated carbon was the best way to do it. And we got a reduction of the metals also by 60 to 99%. It doesn't work for all metals, but many of them. But you don't have to be careful not to dose too much, because otherwise it gets too alkaline, and then you can have negative effects. And the pollenite also works as a good carrier material, making it easier to place it in the field. We use a model called the CAPSIM model, and with this model we're able to predict how long lasting a treatment would be. And we could see that if we add a cap with about 1 to 2 centimeter of this sorbent material in the outer harbor of Oskarshamn, it would be efficient during about 30 years without having to do anything else. And the cost calculation shows that the method per square meter is about 10 or up to 30 times cheaper than dredging. So that is also very interesting. So the take-home message, don't go into the details of what we found in the harbor, but you'd have to trust me that I tell you that we found in the sediment of the Oskarshamn case that even if it has a lot of contaminants, they were not so toxic, meaning they were not so bioavailable. And it questions actually the whole 630 million treatment that was done there to dredge the sediment. And we're thinking, as a take-home message, when the toxicity risk is low, maybe it's better not to dredge, leave it as it is, or maybe do this thin layer cap treatment. And we are now at a time when we need to do more of these pilot tests in the field to test this. And we just got funding now from the Swedish EPA, together with the Borås municipality, to do actually a future pilot test in the Visken River here, beautiful river that was heavily contaminated by the textile industry. And what we will do in this case, we will work together with Vatten Research, Water Resource, a company here that has developed a special boat. You can read more. I have a few of these brochures here if you want. And this boat, you can have different sizes of them, and you basically pump down this slurry of activated carbon and pollenite down on the bottom with adjustable nozzles, and you can, with the GPS system, know exactly how much you added. That concludes the general presentation. I just want to give a few more slides about what Divya was supposed to show. And what this study with Divya is an example of a type of experiments where we do not with capping, but just with looking at contaminant effects. And this is a really, really nice study. I think the best paper that I've done so far together, thanks a lot to Divya. And where we actually could show some things that we often only have had theories about, like how is the tolerance of microbial organisms increasing in already polluted sites. So what we did was to, I skipped this one. That was just why are microorganisms important. I just tell you that they are very important. And it's because they do a lot of functions. You can imagine that when you have nutrient recycling, you have also the degradation of different pollutants like oil, for example. So the bacteria are very, very important in our system. They're also the first ones to get in contact with the pollutants that come on the sediment. So what we did, we took 40 sediment cores from Askö, a clean area, and 40 sediment cores from the Oskarshamn harbor. And you see these guys, the emoji here, this is supposed to show the guys who are the bacteria that are the tough guys. They have been toughening themselves because they've been exposed to copper during like 50 years. Whereas in the Askö area, they have not been exposed to copper to the same extent. We took cores, we brought them to the lab. We had a really complex experimental system that allows us to keep and do an experiment in the lab where we also can maintain low oxygen conditions in some treatments and normal oxygen conditions in some other treatments. So we wanted to test basically environmental factors, both the copper contamination and the oxygen conditions, because the oxygen will act on the bioavailability of the copper. So, and what we measured was what we used new techniques where you actually extract the RNA and the DNA out from the sediment. And from this, it's like when you do detective work and you get like a DNA analysis, we do that for the sediments. But here we also do it so we can measure not only who is there, what bacteria is there, but also what gene are active or are inactive. And we looked at the genes that are responding for metal tolerance. And a huge data set of thousands of pages in Excel done with this sequencing. And we want to understand how the sediment microbes respond and adapt to pollution. And what we measure many things, but as a summary thing, one of the novelty that we did, you have to have threshold concentrations. When you go, you want to know how much arsenic can I have in my water, you have a threshold limit. And that's usually, these limits are called EC10, value environmental concentration 10. So here we measured, but in a new way, how you measure EC10. And we measured this EC10 value as a reduction of the microbial diversity. And what you can see here that is high concentrations are basically meaning that you have a greater tolerance, whereas lower values, sorry, the contrary, these are the higher values. They're high tolerance, whereas these have a lower tolerance. So what we see is that the microorganisms in Oskarshamn, they were actually 2 times more tolerant for copper compared to the one at Askö. And the other thing we see is you compare this oxic and hypoxic, you see that the values are about 1.5 times higher also under hypoxic conditions. So why is that? It's because the bioavailability of the copper is lower under reduced, under hypoxic conditions. And then this indicates that we don't have to stir us blind and just look at the concentrations of contaminants in the sediment. We have to understand what is the pollution history, what is the oxygen conditions in the bottom, how long time have the animals or the microorganism been exposed, because they will be more or less tolerant. And what this means for taking sediment decisions, well, it means that we have to look at the actual ecological impact. They will depend on the environmental conditions. And so we look, for example, we have to consider the pollution history, the oxygen conditions, the bioavailability of the contaminants, the ecosystem responses in general. And disturbing sediments like you would do in dredging, that will change these conditions. It will actually increase the oxygenation of sediments, so it will make many contaminants more bioavailable. So that's another reason for being maybe careful with dredging. So the key takeaways are that high concentrations of pollutants, like they were in the Oskarshamn harbor, are not necessarily mean that it's a high toxicity risk. So we need to do some biological investigations before we do any remediation. To be harmful, the contaminant needs to be bioavailable. So if they are there, but they're really strongly bound already to the sediment, they will not be taken up. They will not constitute a toxicity risk. So again, we have to look at the bioavailability rather than just the toxicity, the concentrations. The bioavailability will decrease with time, and that's the case in Oskarshamn. We had 50 years ago, so most of the pollutants are really strongly bound to the sediment. So we found that only actually 5%, or even for dioxins, was only 2 to 3% of the dioxins that were bioavailable. All the other ones, they were there, but they were not constituting a risk. Dredging is generally not a good option. Dump sediments, during dredging, sediments are reoxygenated, metals are more available and can spread to other areas. Thin layer capping with sorbent can be a better alternative. And as organisms adapt to the sediment, it may be also better to leave the sediment in place. But of course, we cannot just continue to emit contaminants. We have to reduce the pollution, and we should not dump, like you were saying, Ellen, in Konungsfjärden or at some these type of places. That was it. I want to, you can, with the QR code, you can download Divya's paper. And I would like to thank you all for your attention. Thanks.
Wow, very nice. Come.
Here we go.
Okay, guys. I think that was a super good overview and also some really good insights. And as always, we open the floor for questions and also online. And Gun will go around with the mic, and I will have the privilege to start, as always. Okay, so sometimes you do have to move sediments. Maybe you have to make it deeper for a harbor or something.
Right.
Can your method with this activated coal be useful also then? Like you start with that and make it less available when you dump it somewhere, or? Yes,
you were suggesting that you could treat before dredging, actually, to this has not been tried yet. What is usually done is instead that you dredge when you have to dredge, and then you do this method maybe as a post-remediation method to stabilize after dredging. Sometimes they use also other materials like sand or clay that they would put on top after dredging. Then you could amend this clay with activated carbon, and it would make it more effective, because the clay itself will actually not help at all. After just 1 year, the effect is gone. But this, as we see, can stay maybe for 30 years. So not before, but I would say after.
Okay. No questions. And you talk a lot about bioavailability.
Yes.
Can it be changed? We are moving through to something warmer, maybe something more acidic environment.
Yes.
Would that make them more available?
Yes, so the climate change that can, due to a warmer climate, what it often does is that it creates more storm events, and then you will have more resuspension. So what we feared in this case with Oskarshamn Harbor also was that these deeper areas that are not resuspended that much today, but with time maybe there will be more storms, and then you will have more resuspension. And this is the case everywhere. A good thing that I did not have time to show here is actually we were surprised, because just doing resuspension studies, one would think that yes, will release more contaminants to the water, and it will be more toxic, but we didn't see that. For many metals and also many organic pollutants, when we did some small experiments in the lab with simulating boat propellers, we could see that actually many times the resuspension decreases the toxicity, and that's because what is toxic is usually the dissolved contaminants in the water, not the particle bounds. And what happens when you resuspend is that some of the dissolved contaminants, they will attach to the particles and then get back to the bottom. So, meaning resuspension is not necessarily everybody's afraid that we should not dredge here because in this harbor, because it would be super dangerous. But I'm saying that no, not all the time. You have to study that by case. But you have the problem with oxygen availability that could make some metals available. So it's like a trade-off, one towards the other.
Wow, complicated. Now it feels like, if you do dredge and dump, then it would be less available. But
yes,
that could be, depending. Yeah. Okay.
But before everybody asks, I just want to say one thing. That is, when I started to do this work with sediments, I thought this was the most important thing ever. And that's because that's where all the contaminants go. It's 100 times higher in the sediment than in the water. But now, 30 years later, I'm starting to think, what did I do? because we actually find that many times these sediments maybe are less dangerous, less harmful, less toxic than we think. So I want to give a message of hope that we always see that things get polluted and it's really bad, but maybe it's not that bad as it looks. We have really to look at this bioavailability and to see under what scenarios can they be toxic or not. And
also that at least microorganisms can adapt.
Yes.
And also fish, we've heard sometimes.
Yes.
Yeah.
So we too, in 100 million years, humans, we will be resistant to PFAS, to dioxins, everything. So the industry can tell us, toughen up, you guys, just cope with it. But no, so we should not use that as a pretext for not emitting contaminants. Yeah.
Okay, cool. Okay, we have a question.
Hi. Thank you for the great talk. My name's Lucas Winkler-Prins. I'm a postdoctoral fellow at physical geography at Stockholm University. My question is about the oxic or hypoxic conditions. And do you have a sense of the time scale? If dredging kind of re-oxygenates the low water column and the sediment, and then that sediment is then replaced somewhere, do you know how long? I mean, I would think it might go fast, but maybe it's long. How long does that process
I'm not
take?
sure I understand the question. Do you mean how long time it takes to re-oxygenate the sediment?
Or how long does the re-oxygenated state persist?
Oh, how long the re-oxygenation stays before you start?
Yeah.
That will depend on the condition. If you have a lot of organic matter in the sediment, that often, even if you just make a small resuspension, the oxygen will be really rapidly consumed, and it will become soon hypoxic or even anoxic. But if you have a more coarse sediment with less organic matter, then I would say the situation could persist for much longer time. So you have to see what type of sediment you have, and yeah, that's what I can say. Yeah. It will depend.
Okay, we have next question here. Yes, go.
Thank you. Helen Lyman from Baltic Waters. I have a question about the economic trade-offs. I think that will be really important for decision makers. What is more expensive, to dredge or not dredge, to treat with biochar, or do you have any answers about that?
Yes, so when we do these experiments, we buy activated carbon, or we have a collaboration with different producers like Jacobi Carbons, who's been helping us a lot. And for us, we think that the activated carbon is quite expensive. Then having like a few tons, you have to pay maybe $1 million for getting just the activated carbon to the site. So I thought this is quite expensive. But comparing the dredging costs that they did in the inner harbor and looking at the entrepreneur time and everything, we find that this is the numbers that I cited here. We've calculated that the per square meter, it costs about 10 or up to 30 times less than dredging. And that's because we don't have to take care of the sediments, transport them to another place, not build another landfill or put them on the landfill. And it's more rapid also. You can do a thin layer capping maybe in a week or two, whereas the dredging operations can take years. So it's much cheaper to do. That's one of why the USA they started to do this type of capping treatments more and more, because it saves a lot of money.
I think we have a question from the...
Yes, from the online audience. This work?
Yes.
Have you looked at seagrass, such as eelgrass, and how they are affected by TLC? So.
No, I have not. There are some papers that have done that, and they are effects, especially with the powder activated carbon, that can cause effects on some plants, and probably it's due to effects on bacteria that are interacting with the plants. But for the coarser granular activated carbon, no negative effects on seagrass or other plants. And that's actually, we use sometimes biochar to enrich our soil to make a better condition for soils on land. So there's no a priori risk to think that the granular activated carbon would cause problems in plant.
My name is Henrik Svedeng. I have a question about other types of sediments. For instance, at pulp industries, paper mills, a lot of organic sediments, and I think there is a problem with the bioturbation that has increased lately and activated different toxins. Have you any suggestion? Should that kind of organic sediment be dredged, or?
Are you, for example, talking about sediments outside of paper pulp industries?
Yes.
Yes. So there, some of my colleagues have tried that, and it's very difficult because often these paper pulp sediments are super liquid. So it's hard to know where the sediment starts, where the water starts, and it's hard to put a kind of thin layer on top, so it would just sink through it. And you also have gas evolution comes up that can also, for the powdered activated carbon, it just made it not reach the place where we wanted to place it. There are some places in Skellefteå, for example, estuary, where a group has recently done a study with activated biochar. Unfortunately, they used the powder activated carbon, and they used a lot of it. So in terms of retention for the contaminants there, it's been working very well, but they have not measured the effects on any fauna. And I would expect that it has a negative effect on the fauna too there. They say that we should do it, but they haven't done it.
Can I follow up on that? Because now it sounded like animals or microorganisms, they can adapt. But we know that these pollutants are also spreading, and that maybe there come fishes and other animals who eat these, and they spread it through the environment anyway. So even if the animals who live there, they can adapt, but it will still be a problem, right, in the system?
Yeah. I mean, when I'm talking about the bacteria are a good tool to work on because their regeneration time is so fast. So this type of experiment we did with the copper, we could do that in 1 week. But for benthic fauna, you would need to do it over several years, and for fish, you would do it even longer. But the mechanisms would probably be the same. That's why we're thinking that we are actually having probably same type of genetic adaptation to our environment, and that's why we are who we are today. We are evolving all the time. But it's just the pollution goes too fast for us to have time to adapt to it. So we need to slow down the pollution to give us time to adapt to it. Yeah.
All right. Yes, we have a question here.
My name is Niklas Vakeus, and I'm not a researcher. I'm just a normal person. You said that it's not so dangerous, the pollution, as you thought in the beginning of your research. So how many municipalities in Sweden, for instance, are thinking about cleaning this today?
It's a very good point, because now we've been doing with the SGU, the Swedish Geological Survey, and the Swedish EPA, we've been doing databases to see all the hotspots that we have in Sweden, and several of them have already been excavated on land, and the Oskarshamn Harbor was one of them that was dredged. And the idea is that we want to meet this environmental target of a non-toxic environment, or giftfri miljö, yes. And giftfri miljö is a contaminant without toxins. Non-toxic environment, I think, would be a better goal, because that would mean that we can probably leave many of those that have been flagged if they are not, if the contaminants are not bioavailable, and then they will not be toxic. But this is difficult because measuring the concentrations is straightforward. Chemists know how to do it. Measuring the bioavailability, you have to have biological methods to do it, or different types of passive samplers you can use, but they're not always reliable. So. But my recommendation would be to revisit all these sites and really decide on a case by case, not only based on the total concentrations, but based on maybe do some complementary analysis to see how bioavailable is it before we decide to dredge it. And we could have saved probably some operations if we had known that earlier.
Okay, it's time to wrap up. And my final question is always this if you have the power, what would you do? It almost sounded like you already answered it, but
Yeah.
okay, so what's the next step? How can we be better at taking care of these sediments?
Well, I would say I'm a little bit daring today because I say it works and so on. We still need to do a little bit more pilot tests, like large pilots, not only in aquaria. And that's what we will do in Viskan. So in Viskan, it's about 70,000 square meter of sediment that will be treated. So it's big aquarium. There is also a new environment. It's a freshwater environment. You have it's not a very fast running river at those places, so it's more stagnant. So we think the material will stay there. But there are more studies that need to be done. I think we should do some large-scale pilots and maybe to do it together with Stockholm Stad for the Konholmsfjärden, that where they plan to do it. Maybe we can stabilize here instead on site a little bit. So I'm going to talk with them, and I hope some of you follow this path, and because I think we can. These methods are really promising, and my former PhD students, Robert and Johan, who are here somewhere, without them this would not have been possible. So. I'm simplifying things today, of course, but you can look at our papers. They have 2 PhD theses on that, and we have 2 reports coming out from TUFO and from the Swedish EPA also coming out now, and hopefully before the summer.
Okay, great. Thank you. And thank you all for coming. And thank you for you joining online. And I want to tell you that the next breakfast is on the 22nd of April. We will talk about the movement of nitrogen into the Baltic Sea. Some new research coming up, so that's going to be exciting. Yeah, thank you for today. Go out, save the world as always, and see you later.
Thank you. Thank you. Gjorde vi bra.